Hi, Tarun, can you hear me? Hi, Tarun.
Yes, Ashok. Yes, Ajit.
Okay.
Yeah.
Okay, perfect. So I think we can start without thinking much. What do you guys say?
Let Ashok share the screen first and see if it's going well first.
Yeah, definitely.
Yeah, hi Ajit and hi Tarun.
Hi.
Yeah.
Hi. Bye.
Host to disable participant screen sharing.
Oh, so sorry. So sorry. I think so. I'll just kindly give the screen sharing. Okay, given.
Yes, it is fine now.
Yes, it is coming. It is coming. Perfectly fine.
Recording in progress.
You are using two devices as of by.
Tarun, I think Tarun is using both.
Are you using two devices?
No, now I left one.
Okay, great. So let's start. Let's not wait much. People have joined. There are around 100 participants live in YouTube, and they're joining because it's just 4 minutes. So they all are joining. Okay, so guys, good afternoon, and you're welcome to the workshop on the importance of SAS and SPSS in research and industry. So the basic purpose of this workshop is to make everyone aware that how important nowadays these analytical softwares are. Whether you are in the pharmacy field, you are in the engineering, you are in the management, you are in the taxation, you are in the law firm, or anywhere you are, you need these analytical firms. You are in the finance, it is must to use such analytical softwares. There are there are multiple other softwares, but SAS and SPSS are one of the most used, and you must know that from, as we all are almost over 80% of the population here, 80% of the audiences are from the medical and pharmacy and nursing background. We can understand how much these softwares are useful for us everywhere from the scratch. When you start your research, from the sample size calculation to the final analysis, or the subgroup analysis, everywhere you need SPSS and SAS. And SAS is the official software for some government organizations also, and for FDA. This is the official software. They don't use any other softwares than SAS, right? That's why it is very, very important. And you must all know why it is important, why you should learn. And one more very important thing for everyone, now recently government of India in 2020 and 2021, they have launched the new education policy, NEP. Okay, so in that new education policy, they have emphasized that everyone should learn the skill development programs. Everyone should have that the skills, the practical skills, which are very much necessary. Why these are necessary? Because these curriculums are of course they are changing, but in few of what we say in few of the programs, they are still following the old syllabuses, old curriculum. So in that, they don't focus on the new learnings, they don't focus on the skill development program. programs, they are just, teaching theoretical classes, and there's, those traditional practicals, and, in the laboratories, and, you can, work on those things. But these softwares, these skill development programs, these tools are very important, and these are not studied, or these are not, being teased or being taught in the colleges, in the institutions, because these are very, very, very skilled teachers. Also, you need statisticians and, the experts who are working practically in the market, who are working in the colleges, in the specific colleges like, Institute of Statistics, or, biostatisticians. You don't get these people easily, right? And every college do not have the, research setup area, or, they do not have those, things in their colleges, right? So that's why. And for students, from the bachelor's, it starts. From the bachelor, it starts the use of, technical softwares, or, the use of these tools. Because, when you are in the final year of your bachelor of pharmacy, doctor of pharmacy, in MBBS, and, when you are trying for, some research, you want to write some paper, or you are, you are planning for the pharmacovigilance industry, you are planning for, regulatory affairs industry, you are looking for the data management industry, you need to have the hold on these softwares. Nowadays, our as I meet multiple interviewers, multiple, recruiters, what they say is this, they always tell me, like, these skills are not well developed in students, these skills are not well developed in even, the faculty members who are teaching them. And, they are just teaching that old syllabus books and old syllabus, problem solving, these things, but they don't focus on what actually industry needs, what actually, the people need, what actually, the student need to crack into the industry. That's why these tools are very important to, understand what are the uses of these tools, and why we all need these tools, right? So today's workshop is focused completely on these two softwares, two tools, two statistical analytical tools. One is SPSS and one is SAS. Okay, so to discuss SAS, we have Mr. Ashok Chakravarti with us. He will be discussing about SAS, like utilization of SAS and how it is useful, and he will be telling about us the telling us about the basics of the SAS programs also, and he has his mostly he will be sharing all the things why and where needs in the market, and what are the fields where we can apply SAS, and then I'll be taking care of the SPSS after his talk. Okay, so this is a long, I will say the marathon workshop for 3 hours, and it is Sunday. We are engaging you, but I'm very sure that it will be very fruitful for all of you. Okay, very, very fruitful for all of you, and you are going to get the best out of it. Best out of means best out of it. Okay, and yes, it is. You all will get a certificate, but for that you have to stay with us till end, because just 30 minutes before the workshop, we will be sharing the feedback form. And then that is not actually feedback form, that is kind of attendance form, and you will get all the it will get like you have to get the certificate after that. And one more very important thing what we are going to discuss today, we are we have launched our SAS course. As you know, this is in the registrations are opened, and there are only 6 seats remaining in our SAS course. Only 6 seats. We always take, like very less seats are available, because we, go for the personalized trainings in SAS programs for everyone. So the program is almost full. Only 6 seats are remaining, and today's workshop attendees, all the attendees will get a chance to register on the discounted rate. Okay, the official rate what people are paying for SAS course in our in CliMed is 6,000 rupees, 5,999 rupees. Okay, but today they have the special discount prices, so stay with us. That will be disclosed after Mr. Chakravarti's talk. And one more important thing, guys, if you go to the SAS base or advanced, see, in this our course we are teaching you SAS basics and SAS SAS base and SAS advanced, both the things. There are 24 modules only at this reasonable price. If you go to the market, you have to pay 20,000, 25,000, 30,000, and certainly people are charging 50,000 also. And here you will get 99% practical trainings, 1% theoretical things to understand the concepts of the practical. Okay, you will get 99% theory, practical classes from Mr. Ashok Chakravarti in SAS. Okay, so stay with us, and we are launching our third batch of SPSS also today. We have successfully done with 2 batches. We are launching our SPSS third batch, and only for today you will get the discounted prices on SPSS only if you are going to register today. So stay with us and learn well. This is not discounted, and all the things are secondary. First thing is you are going to get a lot of knowledge from Mr. Ashok and from all of us who are going to discuss these SAS program and SPSS program. So I'll not take much time because Mr. Ashok is waiting. So thank you for joining everyone, and stay with us and learn well. Thank you, and over to you, Mr. Ashok. Thank you.
Thank you so much, Adijit. Just I will share my screen. Let me know that whether you can see the screen or not.
Yes, it is visible. SAS is visible. Yeah, SAS. Yeah.
Environment. Okay.
Yes.
Yes. Good afternoon, everyone. This is Ashok Chakravarti. I was working as a statistical programmer and have an 3+ years of experience in industry. And in today's session, we're going to discuss what is SAS, what was the importance of SAS, and where we're gonna use this SAS. So just before going into that, I will show you. Can you see the screen? Yeah. Okay. Coming into the concepts that what is a SAS, we will go through the things like what is clinical trial process, what is SAS, the roles and responsibility of the SAS program, what are the topics going to cover, and the future of the SAS. Because everyone, if they are learning some course, they will be in a position that what would be the future if I learn this course? What would be my career if I choose this course? Everyone is having some sort of doubts in their minds. So I just gonna clear what are the things that are present in our minds, and I will clear all the doubts what you are having. Okay. Before going into the thing, like you all know the things that you are from the science background student or you are from a non-science background student, anything it might be. So every person who is a degree holder or a person who is from science background or non-science background can everyone learn the SAS, because this is the easiest language. Okay, the SAS is the easiest language that is present in the market. This is the 4th generation language. So might be that pharmacy students or the science background students are aware about the clinical trials. So we are having 4 types of clinical trials, that is phase 1, phase 2, phase 3, and phase 4, that is post-marketing surveillance. So why we are conducting the clinical trials? What is the necessity of the clinical trials here? How the SAS is correlated with that of clinical trials? Because you are saying the SAS software and you are saying that clinical trials, how we can correlate both? So you might be having something. So science, what this is clinical trial is relevant.
So I'm sorry to interrupt. Can you make it full screen, sir?
Yeah, sure. Yeah, is it
Thank you. Thank you.
Yeah, you might be thinking, you are saying about clinical trials and you are saying about the SAS software. You might be thinking the software is used by the non-science background students, like that you will be thinking like that, or the BTech students will be using the software or technology or the tools they can work on. But that is not, it's a false perception. Why? Because for the science background students or the science students, we are having a software that is relevant to that of our technology. That is nothing but SAS. So how we are going to use that SAS and how we are going to merge the SAS and clinical point of view. So I want to discuss the thing in today's session. So suppose, just a second. So before that, if we are going to suppose drug. So one drug molecule you are going to introduce into the market. If you want to introduce one drug molecule into the market, what are the things that you need to take? You need to get the permission for marketing. So for getting the permission, to whom you will approach? If you are selling the drug in the India point of thing, you will be going with the DCGI, Drug Control General of India. Or if you are going to market your drug molecule into that of US, you need to get the approval from FDA. Okay, from the FDA you need to get the approval from to deliver your product in marketing into the US point of thing. You will go with that of FDA, and for India point of thing, we can go with that of DCGA. So why we need to get the approval? For approval, what are the things we need to follow? Okay, suppose a drug molecule X you need to place in the market. So for that placing that particular drug into the market, we need to conduct some clinical trials. Why clinical trials are necessary? Why? Because you need to say that my drug is safe, and it is not 100% accurate, but you need to say that how much extent your drug is working on to that particular disease. That you need to get some relevant information. That relevant information should be specified, and that by looking into that relevant information or the documents what you have submitted, from that you will get an approval. So before going into the approval process, what you need to do, suppose you are going to implement this drug XX into the market. For implementing the drug XX into the market, first you need to approach a CRO company, that is contract research organization. In that contract research organization, what they will do, they will analyze, okay, which molecule is this? They will analyze that one, and they will perform those things onto the animals. So that is IND. IND filing they will do. Investigation new drug. Investigation new drug is done onto the animals. Why we need to do or perform the things on the animals first? Before going into the human beings, we need to assure that this drug is safe to work on to the animals, human beings. So in that point of thing, you will be going with that of IND filing. So investigation new drug that was done onto the animals. After getting IND permission, you are capable of doing the same clinical trials onto the human beings. This on human beings, we will be having different classification. That is phase 1. Is phase 1. You might be knowing everything. Phase 1. After that, you will having phase 2, phase 3, and phase 4. That is post-marketing surveillance. So after that, what you will get after completion of the all the clinical trials, you will get some relevant information. So with that relevant information, what you will do, you will go with that of submit the relevant documents to the FDA. So after submission of the document to the FDA, FDA will give a permission for the approval purpose. So this is nothing but NDA, we can call it as. NDA is nothing but new drug application. After getting the approval from the FDA, we will place that drug molecule into the market, and we will go for our business point of view. So this is the thing what it is happening. This is the clear-cut picture from where the initial stages to submission level. So in between, what is the purpose? So this is fine, sir. We know that for implementing a drug molecule into the market, we need to follow all those things. That is nothing but what was the drug molecule, that structure point of thing, and that where it is acting, what were the criteria thing, everything we need to have, and we need to perform an IND point of thing. And after that, we need to perform the same thing onto the human beings. That is the clinical trials. We are having phase 1, 2, 3 phases of studies. After getting some results from or getting some documents relevant to that from the results of that thing, we are going with that of FDA submission. After FDA is scrutinizing all the things, and we are getting approval. So we are going into the market. This is the scenario we know, sir. But where the SAS is working in between. So where the SAS role is playing, SAS programmer playing a prominent role, where we can use the SAS here in between. So we haven't found any such criteria what you have specified. Okay, this is the front end process. What is the really the back end process is happening here means, suppose you are going that of drug molecule. In the drug molecule, we are approaching is one CRO company for conducting the clinical trials. Here, what will happen after IND submissions, you will get to approval to work on to the human beings. That is nothing but that is clinical trials. That is phase 1. Suppose phase 1, phase 2, or anything else. Here, what will happen is, if you want to conduct a clinical trial, okay, if you want to perform a clinical trial, you need to have some relevant documents. Why? Because you need to have some guidelines. What was the dosage you need to give? What was the where should the process has to be happened? How much patient should be taken into the trial? Which type of patient should be included in this trial? Like that, we need to have some relevant information that will be specified in protocol. So protocol is the document that is required for the clinical trial. This protocol contains all the relevant information relevant to that of clinical study. So how the study should be happen, when it should start, when it should end, how many people should be included. Okay, what was the disease condition of the patient should be included in this clinical trial. The age of that particular person should be included. The race, ethnicity, anything, everything will be specified in this protocol. For this supporting document, we are having SAP, and for this thing we are having protocol. We need to have SAP, we need to have, and also we need to have ACRF, annotated CRF. Why this ACRF is needed? ACRF or the CRF, what we will call it as. This is CRF is used in the specific sites. Wherever the clinical trials are performing, the CRF will be present. Why the CRF is needed? Whatever the information that is present in the protocol, that relevant information will be shortcutted and that will be specified in the CRF, case report form, we will call it as. So whatever the tests that are performing, so that relevant information will be captured onto the CRF point of thing. Suppose if you are working on one patient point of thing, suppose you think that patient ID is 101, subject number is 101. For this subject, what you have collected, we have collected his age, race, ethnic, and also we have taken the birth date, and also something relevant information we have collected. Apart from that, we also have collected vital signs relevant information, and also we have collected laboratory science relevant information. You also collected ECG relevant information, cardiovascular relevant information, nervous disorder relevant information. Everything you have collected. So whatever the information you have collected, that information will be specified, or that information will be captured onto the CRF, or that will be captured onto the Excel sheet. Okay, that relevant information will be captured onto the Excel sheet. After this Excel sheet, whatever the information that is present onto the Excel sheet, or whatever the data that is collected onto the CRF, or whatever the data that is collected onto the data sets, we will call it as Excel sheet or data sets. Some relevant information will be collected in data set format, okay, or something will be collected in the Excel. So what is this? Whenever the data is present onto the CRF, that data should be converted either into an Excel sheet format, okay, Excel sheet format, or it should be converted into a data set format. Okay, who will convert this data? So you are saying that, sir, onto the sites we are collecting some relevant information. That information is presenting onto the CRF. From CRF, who will be collecting that particular relevant information? From CRF, case report form, the data what we are entering onto the CRF, or the data what we are collecting onto the CRF in the sites, that information, that relevant information will be captured, or that relevant information will be specified into the Excel sheet format or one data set format. That work will be done by the CDM people, clinical data management people will be there. So what they will do, they will collect whatever the information that is captured onto the CRF or on the sites into an Excel sheet or data set. So they will collect that data and they will convert into this format. Okay. After that, CDM people, what we will do, they will collect that relevant information and they will specify that relevant information into one of the server. Okay. Or database. We can call it as database. So database. From this database, okay, from this database, what we will do, the SAS programmers will come into place here. Okay, the SAS programmer will come into limelight here. So here also the SAS programmer only will do the programming also. This is one step down to that of SAS programming point of thing. So here whatever the data collected on CRF is collected onto the Excel sheet or data set. After that, what they will do, the CDM people will capture that data from the CRF into Excel sheet or data set, and they will specify that particular relevant information into one of the database, or Rave database or Inform database. We are having some database, Linux environment or Unix environment, like that. We are having some environments and some database. From this database, whatever the data that is present in this database, thus this data is called as raw data. Raw data. This raw data is not present in standard format. Okay, this raw data is not present in the standard format. Why we need to have the standard format data? Okay, whatever the data that is present here is not present in the standard format. The data that is present in this database, that is not present in the standard format. So we need to convert that unstandard or the unstructured data into a standard format. Why we need to convert that unstructured or non-standard data in a standard format? Because the FDA persons who who are scrutinizing this data, they need a structured format or a standard data. Why we need a standard data? Okay, for example, here whatever the data present in the database, that is present as a raw data. This raw data, this SAS program, what we are able to do, this SAS program will convert. Okay, they will convert the unstructured data, structured data or non-standard data into a standard format. Okay, and standard format, and they will go for the submission, FDA point of view, submission to the FDA. Why they need to present in standard format? Why means, suppose you are one of the CRO company and another CRO company is there. Some think that XX CRO company is there and YY CRO company is there. So these are both the companies are working on the same molecule. Suppose think that it's one X molecule, they are working on the same molecule, but whenever they are working on the same molecule, finally they need the approval from the FDA. Okay, if you are submit the data in a standard format, okay, if you are submitting the data into a standard format, you will get approval very fast. But you are submit next another CRO company, YY CRO company is submitting the data not in a standard format. In that point of time, what it will take much more time to get the approval process. Why? Because the FDA need to understand whether this is a standard data or unstandard data. Suppose if you are submitted as per the norms that are specified by the FDA, so the process will be very fast. So ultimately what the thing is that we need to get approval fast. For getting the approval fast, what we will do, we will use the SAS programmers. Means FDA, whatever the submission you are doing or whatever the data you are submitting, that data should be present in the SAS format data only. Or we need to program whatever the data that is collecting onto the CRF or that is present in the Excel sheet or the data set that is present in the unstructured data, that should be converted into a structured format, or that should be converted as per the FDA norms by using SAS programming or using the SAS tool only. So ultimately why we are stressing we need to use SAS program or the SAS software, because whatever the value you are submitting, whatever the documents or whatever the relevant information you are submitting, that relevant information FDA is wanting that particular information in SAS programming only. They need that relevant information or relevant data that is programmed in the SAS language only they are expecting. If you are going with it of another tools, they are not expecting. Why? Because SAS is very secured thing. Okay, because you are dealing with that of patient's data, so that patient relevant information should be secured. But if you are going with that of Python or some other tools are present in the market, those are not that much licensed. Those are present in open market. Okay, it's an open market tool, we can say that. But SAS is secured. It has a license, certified license point of thing. So if you want to get one the license for 1%, it costs around 5 lakhs. For the SAS point of thing, it costs around 5 lakhs. So that's the second scenario. But whatever the data you are capturing or whatever the data you are using through the SAS software, that is secured. That is why we are having that much cost for the SAS point of view. So here coming to this, whatever the data you are calculating or whatever the data you are capturing, that data should be reshaped, that data should be analyzed, and that should be submitted to the FDA point of view. Whatever the data you are capturing, ultimately you will submit like table, listing, or figure format only. Okay, here if you go into this, we are having 3 modules again. What is base SAS? We are having advanced SAS, and we are having SDTM. That is relevant to clinical point of view. SDTM, ADAM, and TLF. So ultimately, okay, ultimately what you are going to submit to the FDA is in this manner only, either in the table, listing, or format, I mean figure format only. Why? Because here whatever the data you are collecting, that is the data that is collected onto the CRF, that is converting into an Excel sheet format or a data set. From this, what the SAS program is doing, he is performing some operations onto that data. So here we are having SAS functionality. What is that? SAS functionality. So SAS functionality is nothing but first point of thing, data access. Next point of thing, data analysis. Data management. Data reporting. This is the process we follow. Whatever the program you do, whatever the things you perform in the SAS environment, you will follow these 4 steps. First of all, what we need to do, we need to access the data. So we cannot use SAS programming, or we cannot perform SAS operations anywhere, or in Excel format or text format or any format. We cannot do that operations. We need to perform the SAS operations only in the SAS environment only. This is the SAS environment, how it looks like. This is a SAS 9.4. The latest version of the SAS is SAS 9.4 we are using. Okay, whatever the program we write, whatever the things we perform, we need to perform in the SAS environment only. Suppose if I said that the data is collecting from CRF onto the Excel sheet. So if I want to perform, if I want to work on that data that is present in the Excel sheet, so I need to first import the data. That is nothing but data accessing. I need to access the data. For accessing the data, we are having multiple techniques. Okay. After accessing the data, we want to perform some management on that. Okay. You have access to some relevant information. After accessing the relevant information, we need to manage that relevant information, what we have access into the SAS environment. So if you want to perform any operation onto the data, that data should be present in the SAS environment. What is the SAS environment? Okay. If you look into this, this is a SAS environment. Here I am writing on program data DS. Okay. This is the program I have written. If I want to write this program, if I am writing this program in a text format, or if I am writing this program in Excel sheet, it won't work. I need to write this relevant program. For writing this program, we are having certain rules and regulations also. Okay, we need to write this program in the SAS environment only. So after writing this program, we will execute this program and we'll get the output here. Okay, this is the data set. If you look into this, we call it as this is the data set, how it looks like. Suppose if your data is present in Excel sheet, we need to import the data into the SAS environment. And after importing the data, we need to perform data management. So performing data management, we will use some of the techniques like combining data sets, concatenation of data sets. We will use some of the functions like scan, trim, subster, like that. We will use some of the functions and we will reshape the data. Okay. What we are saying, whatever the data that is collected, that is present in unstructured format. So we need to convert that unstructured data into a structured format. Okay. By using which technique? Data management technique. After data management, what we are having? Data analysis. Whatever the data we are having, we need to perform some analysis onto that particular data. For data analysis point of thing, we are having PROC means PROC summary, PROC univariate, PROC ANOVA, PROC G-chart. Some techniques are there for data analysis point of thing. After analyzing the data, we need to report the data. For reporting the data, we will use PROC report procedure or ODS concept, output delivery system. That is client required output. Okay, this is the flow of the thing. In this point of thing, whatever the data you are collecting, that is the patient relevant information. That patient relevant information I will access into the SAS environment. After that, accessing the data, I will perform some manipulations on that. So some management, data management techniques I will perform. After that, I will analyze the data. Okay, how many adverse events are present? How many concomitant medications are there? How many patients has occurred death during this process? So that relevant information I will analyze. I will get some count. So by analysis, what we'll do, we will perform PROC means procedure, PROC freak procedure. So by using PROC freak procedure, I can get relevant information that how many persons has been allowed, how many persons died with this point of thing. Okay, I can get how much maximum dose they have given, how much minimum dose they have been taken, like relevant information I can segregate. After that, what I will do, I will perform a PROC report and I will specify either in a tabular form or in a listing format or a figure format. Ultimately, whatever the data you are submitting to the FDA, that data should be submitted only after programming with that of SAS software only. So ultimately, whatever the data we are submitting, that should be present in table, listing, and figure format. Oh, if you look into this, I have specified what is the SDTM also. SDTM Adam. So whatever the data we are having, raw data, that we will convert into SDTM, study data tabulation model. This is the thing that is specified by the FDA, that unstructured data should be converted into structured format. Okay, this is SDTM level. After SDTM level, if you want to convert that data into table, listing, or format, we need to convert whatever the data that is present in SDTM, we are present that in character format only. But if you want to represent the data in tabular format or listing format or a figure format, we need to convert that character format data into a numeric format. So whatever the data that is present in SDTM will be converted to Adam level. That Adam is nothing, analysis data model. Analysis data model is nothing but numeric format of the data. So after a numeric format, we can usually use this data to present in table, listing, figure format. So here the SAS programmer playing a prominent role. Here from where whatever the data that is present in Excel sheet and submitting to FDA, this SAS programmer are playing a prominent role. By using the base SAS techniques or using the advanced SAS techniques, we are converting that unstructured or the non-standard format data into a standard format data. So for that conversion, what we are having 4 functionality of SAS. That is SAS access management, data analysis, and data reporting procedures we are having. So in these 4 techniques, we can use either with that of base SAS or advanced SAS. Coming to the topics we are going to cover in this point of thing. Coming to base SAS topics, we will be having some introductory point of thing, data statements. Okay, in that we are having program data vector, various data statements we are having. And for importing the data, how to import, we are having in-file statement point of thing. After importing the data, we are having input statement, how to read the data into particular variable point of thing. And attributes, we are having for each variable, we are having some attributes like length, format, informat, name, data type. We are having some attributes. And also the conditional statement, we are having if and where condition. These are the filtration conditions. Okay, these are used on to the observation level. If you want to filter some condition, we will go with that of if and where statement. Combining set, data set, means if your data is present in different data sets. As I said that this is a data set. Suppose if your data is present, suppose the patient information is presented, suppose it is present in different data sets or different Excel sheets. If you want to combine all the data into one data set format, we will go with that of combining data sets. In the combining data sets, we are having concatenation, interleaving, merge, update, modify. And also the functions we are having. What is that? Date functions, scatter functions, and numeric functions. And some of the other statements also we are having. And we are having PROC import, how to import the data. PROC export, how to export the data that is present in SAS environment. For importing the data means other than SAS environment, if it is present in any other environment, we need to import the data. Why? Because whatever the data you are analyzing into SAS environment, that should be present in the SAS environment itself only. If it is present in the SAS environment only, you can use that data or you can utilize that data only. Okay. After completion of all the things, if you want to submit to the client, we need to go with that of PROC export procedure, and we will specify the same thing in one database. Whatever the data we have performed and whatever the final outputs we are producing, that will be specified in the one of the database with that of PROC exporting procedure. And we are having PROC contents. Okay. We are having PROC compare, PROC append, PROC copy, format. PROC means PROC summary, PROC freak, PROC report, PROC transpose, PROC sort, PROC options, PROC univariate, ODS output delivery systems, do loops, arrays. These all includes into the base SAS part of thing. And coming to the advanced SAS, what we are having is we are having macros and PROC SQL. So whatever the concepts we learn in base SAS, that will be reflected again into advanced and SQL. But the usage or the way we are using those base SAS concepts will be really different. So base SAS is nothing but the basic thing what we are having. Advanced is nothing but some more versatile when compared to the base. Why? Because if you write the code in the base SAS, it will take 4 to 5 lines of code. Suppose think if you are writing some code in the base SAS, it will take 4 to 5 lines of code. But the same thing if you are performing in the advanced SAS, it will take 2 lines only. Why? Because we will use advanced techniques there. Okay, if we get the base SAS knowledge only, we can apply those techniques in the advanced point of thing. You might be thinking, sir, why rather than learning into the base SAS, why we can't go with that advanced SAS? Okay. Suppose if you want to write a paragraph, you need to have a knowledge on alphabets. If you don't know knowledge on alphabets, how you are going to write the alphabets? How you are going to write the one content or one sentence? So first you need to have idea on the alphabets. That is nothing but base SAS. After getting some idea on the alphabets, we will use those alphabets and we will convert that alphabets into one sentence format. Like that only, if you get an idea onto the base point of thing, we will have an idea onto the advanced point of thing. That is macros and PROC SQL. In PROC SQL, whatever the techniques we use in PROC SQL, that is very useful. Means it is very easiest way. Rather than base, we can go with it of PROC SQL. If you write the program in PROC SQL, whatever the code you are writing in base SAS is, if your programming is of 100 lines, in SQL you can conclude that the same base code what you have written in 100 lines in 50 lines only. Okay, that is much more advanced. Okay, these are the topics we are going into the base point of thing and also the advanced point of thing. And coming to the, this is the SAS environment. If you look into this is the editor window. We need to write the program here. After that, we are having the log window. Whatever the things you have executed, whether it is correct or not, you are getting any warnings or you are getting any errors or anything you are reflecting, that will be reflected here. Okay, in the log window. This is the output window. Whatever the output you are generated, it will be reflected in this output window. So whatever the program you have written, this program output will be reflected in the Explorer. See, in the Explorer, we are having this 6 default libraries. In that work library, we'll be storing that relevant information, what we are producing here. Suppose I have written piece of code here. We don't know what the real information that is present in this particular program. So if you want to look into that particular thing, what is that present in that, we need to open this data set. This is nothing but data set. If you click onto this data set, we will have some relevant information. So if you see this name, sex, age, height, weight, this name, sex, age, height, weight are called as variables. And whatever the thing that is present, Alfred, Alice, Barbara, or the male, female, age 14, 13, these are called as observations, or these are called as rows. This is called as column. The name, sex, age, height, these are called as column or variables we can call. And these are called as observations or rows we can call. So this is the data set. So if you look into this name variable, we are having, see, attributes, that is name, that label, what is the label for that particular variable, length of that, format, in format, and data type, whether it is a character data or numeric data. Like that only, if you look into this data set, these are the data set attributes. This is nothing but variable attributes, individual variable attributes, if you look into this. And if you look into this, this is the data set attribute. So here we are having, see, what is this name of that, some name is present. If you are having any label, creation date, modified date, data type, this is the data set. How many columns are there? If you look into this, we are having 5 columns and 19 observations. That is nothing but rows. Okay. See here, we are having how many columns? 1, 2, 3, 4, 5. And observations, if you look into this, 19 observations. Okay. This is a SAS environment, what we are having. And coming to the thing, this is the clinical phases, what we are looking into. You might be well known about the phase 1, phase 2, phase 3, and also phase 4 also. And this is the process how we are going to do. So this is relevant to the clinical point of view. From the CRF, the patient relevant information will be calculated, lab information, medical information, and adverse relevant information that will be specified. And from CRF, CDM team will be there. See, this is the phase, how it is progressing. On to the CRF, case report form. The CDM team, what will do, it will capture the data that is present on the CRF. We are having SOP, standard operation procedure, and documents, electronic files. Here the SAS program, what we will do, whatever the files that are specified in that particular library, they will take that the data that is present in that databases, and the SAS programs, what will do, the SAS programs will perform the process like data access, data management, data analysis, data reporting, and those reports will be submitted to the FDA, NDA, new drug application to FDA point of view. So this FDA, what will do, they review and they give for the approval process. So here from CRF point of thing to the submission point of thing, here the SAS programmers are playing a role, converting, managing the data, accessing the data, managing the data, analyzing the data, and reporting the data. This all the techniques has been performing by SAS programmers in SAS tool only. In that of SAS software point of view. This is the overall flow of this. Who prepares the protocol? You have been knowing about who actually the medical experts, statistician, kinetics experts, they will prepare the protocol. And also the SAP also, it will be statistical analysis branch, means how the table should be represented, how the listing should be represented, how the figure should be represented, whatever the content should be present in all those tables or listings or figure. That will be specified in the SAP. That will be specified in the statistician. Means they will be having some idea. So I need this output. Okay, I need this content in this particular table. Like that, they will predefine before specifying the protocol. After completion of the protocol, they will specify that relevant information. About SAS, generally SAS was implemented onto the agriculture sector. First of all, they are used to this SAS software or the SAS analysis was done onto the agriculture phase. Slowly from the agriculture data, it has migrated to banking, finance, insurance, healthcare, and also into the clinical point of thing. And also different things in different scenarios, we are going to use this SAS in automotive point of thing. In many of the sectors, SAS is playing a role. Not 100%, they are playing 10% or 20% of role in all the sectors. If you look into this, just a second. So in this city, SAS giving operations in various sectors: automotive, communication, education, banking and finance, government offices, point of health insurance, healthcare provider, hospital, entertainment, insurance, life science background, manufacturing point of thing, media, oil and gas, retails, hotels and utilities. In all these sectors, in directly or indirectly, the SAS is using. Okay, in all these sectors point of thing. And if you look into this, who has implemented this one? First, Jim Goodnight and Jimar has been implementing this SAS software. Okay, gradually, eventually, it has been migrated to this clinical sector, and rest of the sectors also it has been implemented. So here, see, SAS giving operations in various sectors. If you look into these are the various sectors, but coming to our point of thing, we are using in healthcare, health insurance, and life science background. What was the purpose of SAS? So data accessing point of thing, data management, user interference, it is application development, business solution point of thing, ETL, extraction, transformation and loading point of thing, and analytics, reports and graphic generation point of thing. This is a SAS functionality: data accessing, data management, data analysis, and data presentation point of thing. Roles and other responsibilities. If you look into this, what is the roles and the responsibility we are having? So what is the role of the SAS programmer? How he is involving in that clinical process? So we have seen that roles and the responsibilities also. So coming to the core functionality of SAS in clinical point of thing, that the protocol design, study startup, patient and investigator recruitment, clinical trial management, clinical data management, data analysis, clinical supplies, regulatory and safety compliance, and regulatory submissions point of view. Coming to this, in where the SAS is operating, in pharmacovigilance point of thing, regulatory affairs, clinical operations, that is CRS, monitors, and also clinical SAS and CDM. These are the things. So a science student can opt for any of this. He can go to pharmacovigilance, regulatory affairs point of thing, clinical operations, that is CRS, clinical research associate and clinical point of thing, and a clinical data management, and also clinical SAS. So here we are focusing mainly onto the clinical SAS. Why? Because it is having vast future. Okay, here the salaries are more when compared to the rest of the things. So here the initial package as a fresher, we are having nearly 4.2, 4.5 based upon the company, based upon the MNC's companies. So they are charging around 4 to 5 lakhs as a fresher. Okay, role of the SAS program, what he is doing in that point of thing. The statistician provide the method for the data analysis, implement this analysis method onto the collected data, and provide the study summary tables, graphs, and data listings to the statistician or the clinician to write study report. So what the programmers are doing, they are creating a table listing and figures. Why? For the writing, for the thing to write by the clinical writer point of thing. SAS programmers closely work with statisticians and data management to provide the link between the raw data and the analysis. In few companies, SAS programs also create the database. SAS programs need to write programs to read database data to SAS datasets and also generate SAS format libraries. SAS programs will also write SAS program to clean the database data according to the edit specified provided by the CDM people. SAS plays a significant role starting from defining the clinical study to till regulatory submission point of view. The clinical study report is compiled using the data generated in SAS environment. SAS also play a role in protocol development, randomization process, and CRF designs, adverse event reporting, etc. In this point of thing, it's an elaborated thing. So SAS programmer plays a prominent role by using the SAS programming techniques. So in this, we are going to cover the base SAS advanced and the SDTM Adams and TLF part of. This is relevant to clinical sector. Clinical domains is nothing but SDTM Adams. So if you want to use the concepts onto the SDTM, or if you want to use the concept in Adams or tables or listing figure, we need to have an idea on base and advanced SAS. After getting the idea on the base and advanced only, we can implement those concepts onto the patient relevant information. So before in base and advanced, what we will do, what we will know, or we will come to know the concepts, how they are using, what is the purpose of that particular concept. So after getting some idea on the base SAS or advances, we can use those concepts onto the data that is collected on clinical trials. So if you are having some concept, so those concepts are used to extract some relevant information of data. Suppose if I want to get some relevant information regarding that particular patient, so you can use that base SAS concept or the advanced SAS concept to get that relevant information in the output. So if you want to work on to the clinical data, we need to have an idea on the base and advanced SAS. So first of all, we need to have idea on the concepts, base concepts and advanced SAS concepts. After getting that idea only, we can really go on to the patient relevant information. We can work on to the patient information. Future of the SAS. The clinical SAS program industry in India has seen rapid germination in the last few decades, and the trend seems to continue for the next couple of years due to cost advantage and availability of skilled labor. SAS continued to be widely used programming language. The SAS Institute, who develop, maintain, and license SAS software, remains a strong data and analytic software company. While other tools and programming languages such as R and Python are gaining popularity in statistics, data analysis, and data science, the SAS Institute continued to be a successful company with a strong market share globally. SAS continued to expand its products offering with the new and innovative tools like SAS via SAS Visual Analytics, helping it to remain competitive in the data science, business, and business intelligence markets. So this is the future of the SAS. If anything comes, either R, Python, or any other tool may come in future, but the programming, what we are doing, or the programming, what we are performing, that should be performed only in SAS. We can use Python also in SAS, or we can use R in also SAS, but that is limited to some extent only. In a nowadays, we are using R. That R is used in the figure generation to specify some more color to that particular figure. So the core concept what we are writing, the core program what we are writing, that will be present in SAS programming language only. Why? Because whatever thing we are submitting, that should be submitted in a SAS programming language data only. So FDA won't accept if we submit in another point of thing. So that's the thing. Whatever the things we are writing, we need to write in SAS programming language only. So R we are using. Why we are means we are enhancing the colors. We are producing figures through SAS point of thing only. Writing the SAS program and we are generating figure. After getting the figure, to enhance that figure point of thing, to add more colors or more thing to that, we are using the R language there. To 5 to 10% only we are using R language. So 90 to 95% we are writing the programming in SAS only. Okay. In nearby future also, we will write the program in SAS only. There is no other options. Okay. That's the thing. Do you have anything to share regarding this, or do you have any concerns regarding this point of thing? Are you having any queries? Just let us go through those things. This time stop staring my screen. Okay. Do you have anything? Have you understood anything about the SAS? Tarun, do you?
Yes, please ask your questions. you can write up your questions in chat box, and we can convey to Mr. Ashok.
Tarun, do you have anything to add more to this?
Dr. Tarun, we have Dr. Tarun also. He's also SAS experts, and he is the our director at CliMed as well. So, Dr. Tarun, do you want to add something?
No. Actually, thank you, Mr. Ashok. I think that was pretty well summed up. Just wanted to give the importance of SAS in this clinical sector, and apart from the various opportunities we have in this field, that is, let it be pharmacovigilance or regulatory affairs or CDM. Okay, so SAS itself has a special and unique place in this clinical research sector, without which it is impossible to submit the data. I think that is pretty clearly conveyed to the audience today by Mr. Ashok. And if they have any questions, I think that is now.
Yes,
now
I have one question to Mr. Ashok and Dr. Tarun, both of you. So the course we are providing, actually, I want to add on first, and then we'll go to the question. So, in this course, guys, you will get the training of how you can, crack the certification also. We will give you the course questions. We'll give you the, he will be teaching according to the certification program as well. So you will be prepared to crack the certification, the SAS certification also, and certificate from our side also you will get. That is totally valid for your job profiles, for your applications, for your CV. But if you want to go for the certification, actual, the international certification, this program will help you cracking that certification also. Okay, so we will be teaching that. So, Mr. Ashok, I just want to know how to, go for that certification, and what is the process, and after that, how it will be, how easy it will be for the, participants who wants to participate in our course.
Yeah, sure. Ajit, just I forgot to say that we need to do the base certification. Why? Yeah. Because we need to do the base certification, so that we will specify that I am a certified SAS programmer. So if you are having as a certified SAS programmer, you will be having more opportunity. Your resume will pick more when compared to the non-certified candidate, because all the MNCs who are looking, or the clients who want to work with them, they need the certified SAS programmers. So yes, whatever the course we are teaching, or whatever the things we are sharing, one thing we can say that after the completion of the course, we will give you the required document also for the practice session and everything. And we will also conduct some examination, or the test we will that will gain you more skills on you. On if you are lagging if you write the exam, you can know that where you are, so that you can improve yourself also. We will conduct 2 to 3 tests, and also we will conduct a final test also. Okay, so yeah, if you want to go with that certification, whatever the course we are giving, or whatever the things we are specifying here, that is sufficient for you. That is more than enough. So we will train in that way, in that manner, so that you will be parallelly getting knowledge regarding the certification point of thing also.
Yes. So some people are asking about the TLF. What is the full form, and what is TLF actually, Mr. Ashok?
Yeah, TLF is nothing but tables, listings, and figures. After you are getting some knowledge on base and advanced, then that is the second thing. That is clinical SAS, we will call it as. That is not a part of this thing. That is relevant to the another part. So what are the concepts we have learned? That concepts we will implement onto the clinical data. That is, we will have a project relevant to that. So that is separate. But here, what I have, why I have specified the TLF here means, whatever the concepts we are going to learn, with that concepts, we will convert that data into table, listing, and figure format for the submission to the FDA. Finally, FDA want not the document relevant to that of data. They need it is in pictorial representation or in a tabular representation for easy accessing for them to get the drug approval point of view.
Right.
Yes, just to add to that, what Ashok said. So before we get into clinical SAS, TLFs, SDTMs, and ADAMS, we need basics, right? So that basics is what we are covering in this course. That is SAS base and advanced. After this course, we are also planning for a different course, that is a project related work. We will let you know shortly. Okay. So this base is like helpful for anyone, not just the clinical background people or medical background people. So anyone who learns SAS base and advanced can go into specialization, that is automobile or insurance or banking, etc., etc. Similarly, we are focusing on clinical. So that clinical project and course related data will be given to you shortly. Right now, we are focusing on the basics, fundamentals.
Right now, the course, okay, Kavita is asking if we are providing everything in 5,099. No, we are providing base plus advanced. There are 24 modules. Okay, there are 24 modules. Base and advanced are summed up into the course, and clinical SAS will be different, where you will get all the clinical practices and all the clinical data as well. You will get the live data trainings in the clinical SAS. So it is not the clinical SAS. We are not right now focusing on clinical SAS. We are providing, but not only base, we are providing base and advanced, both the versions of SAS to you in this course in 50099. And for that matter, we have linked, we kept our links in our description of this video. You can just check out the description of this video live program. You can see the description, you will get all the links. So all the links are shared already. We have kept in our data in this description of this YouTube live. You can access the links here by tonight 11:59, and you can register with these prices. Otherwise, the actual price of SAS is 5,999. But now today we have given additional 50%, additional 15% of 6,000. So 900 additional discount to you. So you can just register to this course only in 5099. And then SAS course, now we are launching the SAS course with the SPSS course with the duration of 2 months, again 8 weeks course of SPSS, complete SPSS practical programs, practical training, and that course fee will be 1,999. But today, as you are attending this course, you are attending this workshop, you all will get this SPSS training course in just 1,499. So there is no extra charges for the certificate from our side, Nitish. The international certificate, there is the fees, but we are not charging any fees from our side. This fees is the entire fees what we are getting for SAS base and advanced, and providing the certificate from our side. Our certification is you will get in this fees only. Duration of the course is 3 months. Every week there are 2 classes. Saturdays and Sundays there are 2 classes. Every week 2 hours every evening. Okay, 3 months course this is. Duration of the course is 3 months. Okay, so someone is there is no eligibility to join our course. Okay, if you are in bachelor's, master's, researcher, or any specialist, industrial delegate, anyone can join this course. No need to have any prior knowledge for programming. No need, because we are starting from the very scratch. Smiley, we are providing everything, all the knowledge from the scratch. No need to have any kind of prior. No, this is totally online. Right now, it is not offline. It is totally online, but online only you will get the entire practical. Yes, completely online. No eligibilities. Okay, so I just want to know, Mr. Ashok, is there any eligibility for the certification if they want to go for the international certification?
Yeah, eligibility means they should have a degree.
They should have a degree.
UG or PG, anything. If they need to have a degree point of view, because if they want to enter into the industry, shortly they will ask for what was the education background.
Yeah, education background, correct. Yeah.
For so certification point, anyone can write. A degree holder can also write, or a UG person, or a PG person, or the master, PhD person. Anyone can. This certification is globalized. We can use this certificate anywhere in the global.
Yeah.
This is lifetime validity.
Right. Okay, someone's question is very interesting here. Clinical trials may not be conducted every time, like lifelong, right? So how can we demand for SAS programmers lifelong? So these clinical trials are not only one. There is not only one clinical trial.
Yes.
Sai, there are thousands of the clinical trials, and if I am not wrong, I was reading one report, in last 2 years, in COVID only, there were something 2 million studies are going on all over the world. New studies in last 2 years, 2 million new studies at the institutional level and at the industry level. 2 million study. Can you understand? 20 lakh studies are going on.
And if you see the overall life of a trial from phase 1 to marketing, phase 4, it easily takes at least 10 to 15 years. So one study is itself around 10 years. And you can imagine 2 million studies.
Yeah. And now I'll say next 15-20 years, there is no lack in the clinical trials, because see, COVID is not only that it is related to COVID. COVID has impact on every disease, every condition, every comorbidity. COVID is in diabetes, COVID in cardiovascular, COVID in arthritis, COVID everywhere. And these infectious disease and antimicrobial resistance, thousands of the things are there. So clinical trials, as as well as we have human beings on the earth, there is no lack of clinical trials, I'll say.
If the trial has been completed, they will switch on to the another study.
Yeah, they will switch to another study, correct. And drugs and everything is coming up. Yeah.
Just if you take only COVID, there are hundreds of studies going on. So if you take another background or another antimicrobial,
respiratory, cardiovascular, a lot of things we are having.
Depending
lot
on
of
the
therapeutic areas.
area. Yes.
So
See,
it's an ocean. It's an ocean. There is no end.
Yeah. So only clinical research is an ocean. Other than clinical research, SAS has its application in agriculture, in other medical things, in industry, in finances, banking, everywhere. You just should understand how to use it.
Yes, I have a question to Mr. Ashok regarding Mr. Ashok. Can you just throw some light on the career pathway of this SAS? For example, if someone joins as a fresher, what is the scope in India and abroad?
Whether in India or abroad, the thing is, whatever the programming you do here in India or in abroad, point of the programming will be the same, but the package differs there and here. That's the main. Here we will get a INR, Indian rupees. There we will getting some Canadian dollars or US point of thing, like that. But whatever the work we are doing is, whether you work on to the US level or in India level or Canadian point of thing, the work culture will be the same as such only. But whatever the scope of this thing is, nothing but if you join as a fresher, obviously fresher will be having a designation as clinical SAS programmer or SAS programmer. After that, you will be having statistical programmer 1, stats programmer 2. Then you will be having programming lead. After that, will become manager. So this is the hierarchy. After that, manager will become a director point of thing, like that. Associate director point of thing. This is a scope. Yeah. As I said, that initially we will be starting a career with that of 4 to 5 lakhs of package. Eventually, if you go with that of market analysis or market, as of now in the market, if you are having 2 years of relevant experience, if you are having 2 years of relevant experience in SAS programming, they are charging around 12 lakhs.
Yeah,
that's the huge amount. You can say that no other industry will give you of 2 years of experience or 10 lakhs, 12 lakhs of package. So that is the main advantage of why they are giving this much of amount. Why? Because they need the approval fast. For getting the approval fast, what they are doing, they are taking the source of the clinical programmers, SAS programmers. So they are doing their process very fast to get in the approval point of thing. That's correct. The main. So they are giving huge amounts and they are paying huge amounts to the SAS programmers to get their work done fast. That is the main.
Right. So we have 1 or 2 more questions, and then we'll relieve Mr. Ashok. So someone is asking about the placement assistance. See, guys, we will refer you. We will give you the complete training, and we can refer you. But we are not, if we need, we can try to give from our side. But we are not 100%, assuring you that we are, giving you job, or this program will, give you job right away from our side, or we can help you definitely. We can assist you in getting job, but there is no placement guarantee, frankly, because we are not into the placements, we are into the education. That is our motto. We are providing the quality education. We are giving you the 100% the live training. Live training means you will be getting all the trainings from Mr. Ashok every week. There is no recordings. You will get all the live trainings and all the practical learnings from Dr. Ashok, Mr. Ashok. So there is no we are not providing the placement assistance definitely, but you will get the quality education. You will get the quality training here from our side. Okay, and that's why we conduct these courses. Saturday, Sunday timings will be usually we keep it from 5 to 7, sometimes 5 to 5:30 to 7:30, or sometimes 6 to 8. So usually we start between 5 to 6 on Saturdays and Sundays. So okay. Yes, Sai, our institute is only providing this course in 5,099 today. If you are registering today, someone has the question: do we have to pay entire amount today? No, you can pay in 2 installments also. There is no problem. You can pay 3,000 today and rest later also. That is not a problem.
Just to go through the market assessment, how they are charging for
yes.
Just to go through that also. Just don't blindly join, or don't blindly they are suggesting that.
Very interesting question. Very interesting question. Sai has come up. Official SAS website is offering the SAS course for 75,000 and 75,000 for advanced. Then how could you provide just in 5,000? That is the perfect question, and I would like to answer. See, CliMed, we have started 2.5 years back, and 2 years back we have started educational services, and our main motto is not to just like others. We are not here for earning money, because we have our other things. We have the CliMed is not only educational services. CliMed is CRO. Okay, CliMed is contract research organization. So we are getting enough money from the industry side. Okay, don't worry. So from these courses, we are just charging the technical charges or certification charges, nothing else. That's why we are providing. That's why people also don't believe, but we have successfully completed 2 batches, not only one. SAS, we have successfully completed 2 batches. First was started in April 2021, second was started in July 2021, and we have completed recently that batch. So and all the batches were full. We always try to sum up our batches in 20 people, 20 students in one batch, because we give individualized training to everyone, individual query resolution, individual training to everyone, practical knowledge to everyone. We help in getting you the software also in your computer. If you have any queries, our Mr. Ashok solve it live with you. Okay, that's why we don't entertain more than 20 students in one batch, and there are only 5 or 6 seats are remaining in this course as well. So that's why I'm telling, this is a quality education, not about the money. Okay, they are there are in the Indian market also, you will get this course from 25 to 50,000, because they are professionals just to make money. We are professionals to give quality. We are here to provide the quality. We don't have only SAS, we have SPSS, we have regulatory affairs, we have pharmacy courses, training programs. We have around 150 colleges in India under our MOU, where we are providing skill development programs, and that is of course free of cost or at the minimum cost. Okay. Yes, definitely. Okay, so there are the questions. Okay, someone is asking, Yamini is asking, sir, is it mandatory to be trained with SAS to enter into the CDM industry?
Yes, we need SAS knowledge to enter into CDM industry.
Yes. Others, yes, if you are a doctor of pharmacy 5th year student, it will definitely help, because people, industry, if you are planning for industry or the research, you need such courses. You need these courses. There is Dr. Venu, there is no basic knowledge required, because we are starting from the scratch. We are starting from like how to download the software, how to open the toolbars, and what are the toolbar means, everything, and then we go to the extensive analysis at the end, to the advance. Thank you, Salim ji. Thank you very much. And thank you, everyone. So I think we should not delay much, and I should start, the SPSS part also now. We have SPSS workshop also. And whoever wants to register, please go to the links we have provided in the description of this video, description of this live, video. And if you are registering today, then only, these links are valid only for 24 hours. Okay, only for 24 hours. If you are registering by using these links, then SPSS you will get in 1,499, and SAS course you will get in 5,099. Okay, if you are registering after today, then you have to pay the entire fees, 5,999, 900 extra. Okay, that is because you are joining us in the Sunday afternoon. You are leaving your Sunday enjoyment and joining us and learning with us. So that's why this is a concession for you. This is a discount for you all. Okay, and you you can understand that we have completed 2 batches successfully from our CliMed research solutions, and those were completely full. Okay, and this is the 3rd batch we have started now, and this is also almost going to be complete in 1 or 2 days, and we are starting this batch on 17th. No, you cannot pay the half amount from this. I'll just share the Google Pay number. You can, because this link is for the full amount. This is the Google Pay or Phone Pay number. You can pay to this number half amount, and you can send your details to WhatsApp of this, and course name, whatever the SAS, and your name, your email ID, a phone number, 3 things. Send me on this WhatsApp itself, and pay the half amount to the Google Pay or Phone Pay, anything. This is the method to pay half, who wants to pay in the installments. But if you want to pay the complete today, you can go through the link. Okay, so thank you, everyone. Thank you, Mr. Ashok. And I think, I'm sorry we have delayed you for 18 minutes. Thank you, and we'll see you. Thank you, Mr. Ashok, for joining. Vishwanath sir, no, it is on the weekend, Saturday and Sunday, 2 hours per day. 2 hours on Saturday and Sunday. Feedback will be shared only before 30 minutes of the end, not right now, because our SPSS is also going to start. Okay, so thank you, guys. Tarun, I think Dr. Ashok has gone. No problem. So let's start the SPSS. So, guys, before starting the SPSS, I would like to tell you a little bit about me. I have done my PhD in cardiology from the eminent Kasturba Medical College, Manipal Academy of Higher Education. And, I have published almost 40 plus papers. And, I have a good experience on SPSS, and I have worked in the biostatistics also, and epidemiology. And, there are certain courses what we have, I have also done on biostatistics and SPSS. Dr. Venu, the duration of the course is 3 months for SAS. Okay, then, so I take the SPSS course, and we have completed 2 batches of SPSS also. We always run SPSS and SAS together. The SPSS course was also started in April 2021 and then July 2021. So April batch, we had 150 participants. April 2021 batch, we had the full batch of 150 participants. And July batch, we had 100 because previously in April batch there were too many, so we restricted the batch to 100 people, and it was completely full. Our April batch was full with 150 students, 150 participants, and July batch was full with 100 participants because we have restricted. Okay, and those batch are successfully completed. Even July batch has completed now only. There is, I think, 1 more class is remaining, and their certifications is remaining. We conduct classes on the weekends, and even sometimes in the weekdays also. And in SPSS also, there are SPSS courses 2 months, 8 weeks course, and there are 20 modules. SPSS, there are 20 modules, right? So in the SPSS, you will get the entire things. I'll show how and what it is and why it is important for all the students. See, first of all, let me share my slides so you can easily understand what is SPSS and why it is useful and what you will learn from it. Okay, so yeah, here. So okay, I hope it is visible to you. So this is our the course what I was discussing with you, the course highlights. Okay, so as our SAS course is starting on April 17th, and registration last date is April 10th. But if you are registering any other day, you have to pay 5,999. If you are paying today, you just have to pay 5,099, 900 rupees discount. Okay, for SPSS also our actual fees is 3,000, but we are charging only 1,999. But today, if you are registering for the SPSS 8 weeks class, 8 weeks courses, you have to pay just 1,499. So straight 50% off on the actual fees. Okay, just 1,499 you have to pay, and everything is live. Okay, all the classes are live, and you will get the complete package from data entry to typical test applications in SPSS and SAS both. Okay, and anyone can join: medical, pharmacy, life science students, researchers, professionals, academicians, anyone can join the SPSS, and no specific skill required. Okay, just who wants to advance their skills in the statistical analysis can join any of the course. Okay, so we have as I was just talking about, your links are there. If you want to join the SPSS course, you just have to pay 1,499, but today itself. If you want to join SAS, you just have to pay 5,099 rupees only for today. Okay, and for this workshop certificate, you need to fill the attendance form, which will be shared 30 minutes before the end. Okay, so let me come to the SPSS now. I think there is a lot of information on the technical part or the advertisement part, but now let's come to the SPSS. What is SPSS? SPSS is nothing but it's a statistical software, and where you must have seen this SPSS and where you are going to learn about when you are doing a clinical research in your, right now, if you are a student, if you are doing the research, not only clinical, if you are doing the research and you are going to analyze your data, you usually do in Excel sheet, usually do in Excel, but everyone is not master of Excel, and Excel is not having those tests as well. Excel gives you the basic frequencies, Excel gives you the basic tests. Okay, Excel will give you the basic results, but after Excel, if you want the specific results, if you want to correlate something, you want to relate to correlate two variables, if you want to associate two variables, then you have to go for the SPSS analysis. Okay, so if you are a medical student, if you are a PharmD student, or if you are a B.Pharm student, M.Pharm student, or if you are a PhD researcher, right, and you are doing a research and you want to analyze the data, you have collected the data from patients, from surveys, from anywhere. Okay, from anywhere you have collected the data and you want to report it. Okay, after collecting the data, you want to report it. How you will report? You cannot report the Excel sheet directly. You have to convert into the results. You have to analyze the results. You have to find out the frequencies. You have to find out the percentage, mean, mode, median. You have to find out other things, like chi-square test. You have to apply, multiple tests are there. Also everything. Okay, what is useful and how it is useful in the research. So mainly I am from the clinical background. I'm from the clinical, side. So I will tell you all the benefits and all the applications of SPSS in clinical side. And I will tell you even in the, you can use this in the banking, law, management, finance, commerce, everywhere you can use the SPSS. It is very, very important to get the analysis done. And not only this, you must have seen the political surveys. You must have seen the political surveys. They show before any elections, they show, okay, 50% seats will go to this party, this party, this party, and you will get they will get such percentage of women's vote, such percentage of men's vote. How they analyze it, political parties, and everywhere in the wherever they need analysis, extensive analysis, they need the analytical softwares, and SPSS is one of the best. SPS is one of the best, and SAS is SAS is the official software of FDA, so we cannot say that you don't need SAS, right? SPSS is required everywhere, and SPSS is the easiest user-friendly software. Okay, so SPSS is originally a statistical package for social sciences when it was released for first time. Okay, but now it is like it is used for everything, and you can use for mainly it has been used for clinical research. Okay, so it was released in 1968, and it was developed by Norman Nye and C. Heldi Hull Norman, and they were the political science postgraduate from the Stanford University, as I'm telling you, political science postgraduates. So but now it is most used in the clinical research, guys. Okay, and SPSS is among the most widely used programs for statistical analysis in the social science and other sciences, right? So SPSS has been taken over by IBM. IBM is one of the biggest company of software making and hardware making. Okay, you must have seen the laptop, desktop, and multiple softwares of from IBM. If you are from the clinical side, from the pharmacy background, you must know Micromedics, right? Micromedics is IBM product. SPSS is IBM product. Okay, so this SPSS is now statistical product and service solutions, because it is providing services and it is providing analytical analytical products in every background, every domain, not only in the social sciences, right? So between 2009 and 2010, the premium vendor of SPSS was called PSW, but now it is SPSS by IBM. It is now IBM SPSS. Okay, and the latest version, because they update every time, they update the version. So the latest version in the market is SPSS 27, SPSS version 27. Okay, what I am going to show you is SPSS 22. Okay, SPSS 22, that version 22 has come in 2017, not now, 2017, 5 years back, 6 years back. Okay, we are still using 22 because upgradation and everything is very costly for the software. Okay, I'll show you why it is. So you can see the software 27 version has been launched on July 19th, 2020, and developers, as I told you, there are Bent, Haldai, and Nye. And operating system, if your laptop having the window, it is working everywhere, Windows, Mac, or Linux, everywhere. Okay, and the platform for the software is Java, but you can use in the normal softwares which has the 30-bit or 60-bit. Okay, but these 22, more than 20 actually, more than 20 version of the SPSS V20 or more, anyone anything, they require 60-bit laptop. They are not applicable for the older laptops. The newer laptops, which are actually these newer laptops, also 60-bit has been launched long back, 10 years back, 8 years back. So whatever you have the laptop, if your laptop is after 2015, then you can use the SPSS version 20 or more. Okay, so what is the cost of software? I'll tell you guys why it is very, very important, and why they have this much, and what is the cost actually they charge. SPSS monthly subscription is 99 USDs. Okay, 99 US dollars per month per user, and monthly auto renewal basis. Okay, IBM basic SPSS base version 26 is 1290 USD per year. Okay, around 85,000 per year. IBM SPSS standard is around 1.8 lakh, 1.8 lakh per year, and IBM statistical professional version is around 4 lakh. Okay, 3.8 lakh, around 4 lakh per year. Okay, so that is the cost. So we have the SPSS available for our university. Usually universities, institutes, colleges, they take institutional subscription. They usually pay around 12 lakh per year. Okay, they usually pay 12 lakh per year for the statistical software, and they give the keys to their researchers. They give keys to their faculty so they can analyze their data and their students' data. Okay, so and this is very very important software to use with everyone. And why it is costly, that's why I told you, because it is being used everywhere. It is being used widely, not only the social science and clinical research of banking, management, law, everywhere, right? Now let's go to the use of software, use of SPSS, where you can use it. This is IBM software. It enables educators to teach effectively. It is not only for the students, it is for the teachers, it is for the educators, it is for the mentors. Because if they are good in biostatistics, if they are good in SPSS, practical trainings, analytical softwares, analytical methods, they are really good, and they can get the precise data results. They can get the results what actually the research objective want, right? See, suppose I'll give you the example here, how it helps students and educators. Suppose you are a professor, you are a professor or associate professor, and you have the permission. Now you are just you have landed into that phase where you can take the PhD students, or where you can take the master students to guide, right? MPharm students or MSc students or MTech students, you can get any students to guide. So you have to initiate the research project, right? You have to initiate the research project. Suppose then if it is in the clinical side, suppose I'm just telling you, if it is in the clinical side, you need a sample size. How you are going to calculate the sample size? If you are going to calculate the sample size, you need specific softwares, specific tools, where you have to keep the p-value. P-value means the power of the study, and the previous sample sizes, and the constant, and many things are there what you have to keep in the software. And then it will give you the your sample size, like in how many patients or in how many subjects you should conduct the study, right? So that time also you need SPSS, because SPSS has the easy version to calculate the sample sizes. Okay, it is like it has the inbuilt formula. And one more thing, SPSS is nothing where you have to, this is the first software, this is one of the easiest software, user-friendly software, where you do not have to run any kind of codes, where you do not have to learn coding. There is no code required. Everything is already established. You just have to put your data, refined data. Definitely you have to refine your data, you have to clean your data, you have to put into the SPSS, and just run the SPSS. One click away, all of your results are just 1 second, 1 click away. You need not to run any kind of software. Just select the variables in your data set, select the test which you want to apply, click on OK, and you will get your results. There is nothing more you require to do in SPSS. Okay, guys. So it is very easy, user friendly, and it is very much helping in your learnings. So when you are doing the research, you need to state your sample size, and then you have to conduct the entire research. You have to collect the data, and you put the data in Excel sheets, okay, to refine. And you will, like, suppose you are collecting the data about corona. What are the symptoms? What is the age of the patient? What are the outcomes? What are the treatments they have taken? Multiple things you are collecting nowadays. You must have gone through the surveys. Multiple surveys are being conducted in the society. Every medical student, every pharmacy student, psychology students, everyone is coming up with the surveys. They're like, impact of corona or impact of lockdown on people, impact of lockdown on health, impact of corona on health, impact of corona on cancer patients. When multiple surveys are going on in the market, so how they are going to tell you, that what is the impact of corona on the mental health, or what is the impact of lockdown on the food habits, or what is the impact of lockdown on your exercise habits? How they're telling you, how they're going to check that impact? That impact will be checked by using the statistical software, by analytical softwares, and SPS is one of them. Okay, as per I am in the medical market, medical thing, medical field, or dealing with the pharmacy trials, or dealing with the medical trials or clinical trials, I know what is the use, because everywhere I don't see any other software than SPSS what they use. I'm working in ICMR as a research scientist. I'm working for WIHF as a National Director for Research. I have seen no other softwares better than SPSS, and they all are using SPSS. Everywhere it is useful. Okay, in nursing, in pharmacy, everywhere. So it helps educators to teach effectively if they are pro in the softwares. They can tell you, they can tell their students, you can tell your students, okay, this is the objective and this is feasible to do. This is easy to do, this is easy to analyze. Even after once you collect the data, you will guide your students correctly, what to collect, what to analyze, how to analyze, what test will be applied when you are collecting, when you are analyzing the data. If you are perfect in the softwares, if you are perfect in the biostatistics, the statistics, SPSS works on the statistical methods. SPSS is the statistical software. It is based on the statistics. So whatever research, whether the research is in banking, commerce, management, law, If you want the results, you have to do the statistics. You have to do the statistics. And when it comes to the market, it comes to the medical side or health science field, it is called biostatistics, because it involves the bio. It involves the biological samples. It involves the biological characters like human beings, animals, or any kind of study related to human. I mean, the health science stream comes under bio, right? So it calls at biostatistics, right? There are the different types of methodologies, how to conduct cohort studies, clinical trials, randomized control clinical trial, epidemiological data, collection, community trials. Multiple things are there, right? So everything, if you know these things better, if you are pro in biostatistics, if you are pro in softwares, you can tell your students, okay, this is the best topic, these are the best objectives to conduct, and this is the best way to conduct your study, because you know how to get the results, how you are aiming, and what you are exactly aiming to get. You know that if you are expert in that, and then you can teach your students the same. Okay, you can teach your students, and you can claim your students to do these things. Understand? So it helps also, it helps students to gain critical analytical skills and support more accurate and insightful institutional research and decision making. Yes, of course, you will get the accurate and precise result after these software analytics. Otherwise, see, if you are doing in the Excel, you will get the results, but to a certain extent, to very basics like frequencies, mean, mode, median, those things, sums, and you can get those things. But after that, what? If you want to correlate something, if you want to dig something out of your data. Suppose I have collected thousands of the corona patients. Okay, thousands of the corona patients data, and their comorbidities, their gender, their age, their educational background. So now I want to see how I'll just see like what is the impact of corona on the food habits. Suppose, right? Suppose I want to check the food habits. So there are multiple factors which can affect the food habits. Okay, because there was the availability of food. You cannot go out, or if you were if you are working, then how can you, and if you're working, then you cannot get the food easily, or you have to eat outside, or you have to keep the maid, and due to lockdown there was not possible, multiple things, and you have to restrict your food habits because you are not going out, because you are not allowed to go out. So other factors also related to that, right? So multiple factors were there, right? Multiple factors there. How to correlate those factors, like which factor was most influential on your food habits, right? Those things you have to understand, right? Those things you have to understand. So how do you do that? How would you do that? Actually, you have to go to the statistical software. You will keep all the variables together, you will keep all the risk factors together, and then you will run the software, which risk factor is more correlate, or more correlated to your food habits, change in the food habits, right? You will get it easily, you will get it easily, you will get the accurate results. See, there are five things which are, suppose getting heart attack, normal question for all of you. Like, suppose we are going to check heart attacks. Okay, in the heart attack patients, we are getting what is the risk factors, or what are the risk factors. We all know, like when the patients are getting heart attack, we expect that patient was diabetic, patient was hypertensive, patient was a smoker, patient was patient must be alcoholic, or patient would have been into food habits, their food habits was not good, stress was more. These all are the risk factors, right? These all are the risk factors for heart attack. These all are risk factors for myocardial infarction, right? Like your hypertension, diabetes, or your smoking, your food habits, your sedentary lifestyle. Many things are there, many things. But what is actually significantly related to heart attack, or related to MI or not? How you are going to check it? How you are going to check which one is precisely associated? By just because I am saying, you cannot say that everything is related to heart attack, because it is proven. That's why I'm saying, of course, statistically proven. But for, like, suppose where we don't know, we can just expect, we can just guess. But in statistics, or in medical science, or in any science, guess won't work. Guess actually they don't work. Your guesses will not work at all. You need to have the precise results. You should have the insightful results, where you can get, okay, this was the factor, Maybe it is surprisingly something else, but you will get the exact thing, exact statistical part, exact statistical result that, okay, this was the correlating factor. Oh no, alcohol is not relevant at all. It was diabetes which was causing heart attack. Oh no, it was not, diabetes, it is hypertension. It is high BP, which is relevant to, your heart attack, because it is showing the p-value less than 0.5. P-value less than 0.5 is significant. And for smoker, in your study, smokers have the p-value more than 0.05. That is like suppose 0.6 or 1 or 2 or 0.6 or 0.7. That means that is not significant. So just by assuming or guessing, you cannot pretend, you cannot predict the result. You have to go through the statistics, and then you will get accurate or precise results, like what is exactly relevant and what is exactly correlating with the outcomes or with the objectives, right? So your decision making will be precise. Your decision making will be sharp by using these softwares, by understanding these software, by the knowledge of these softwares. Understand? So now with SPSS statistics, what you can analyze and better understand your data and solve complex business and research problems through a user-friendly interface. These 4 points, I am not saying, these 4 points has been taken from the IBM website, IBM SPSS statistics, what they claim. And this is right, I can prove it because I use everywhere this, okay? And you won't believe about the jobs and all. I'm just in between this slides. I will tell you guys. Statisticians, they charge 6 to 10,000 per hour for your analysis. Statisticians, professional statisticians, if you are going with the research thesis, if you are going with your thesis because you are not pro in SPSS, because you don't have the software, because you don't have the resources with you, they charge 6 to 10,000 per hour, or like per thesis 15,000, like lump sum 15,000. It is just a work of 2 hours or 3 hours. Okay, it is a like boom, lot of money they charge, because they are pro, they are the masters of statistics, they are the masters of biostatistics, because they have the software. They don't take time, they just see, okay, your data is defined or not. They will ask you, refine the data, give it to me, I'll analyze. They will analyze in half an hour, they will charge you 10,000 rupees. Okay, that is the use of statistics and knowing statistics. You can save your money, you can save your students' money, you can save your colleagues' money, and you can earn money, right? This is just a secondary part, I'm telling you. So you can analyze and better understand your data, and you can solve the complex business and research problems through a user-friendly interface, because SPSS is user-friendly. No need to code, no need to go for coding or anything. You don't have to know the Python, Java language, nothing. Just SPSS is fit for everything, because there is no coding. Okay, understand the large and complex data sets quickly with advanced statistical procedures that help ensure high accuracy and quality decision making. Complex data sets, I am talking about complex data sets here. Like you have thousands of variables, not only one. You have 10-15 variables to analyze together. You want to check age together, you want to check comorbidities together, you want to check social habits together, you want to check treatment patterns together, and outcomes, everything. There is no stress. Just run it, click it, you will get the results. Okay, and accurate results. And see, when you just, okay, you want to make a sum of 10 digits, like 5 plus 5 plus 5 plus 6 plus 3 plus 2 plus 1 plus 2, you will get it done in just 5-10 seconds by yourself. If you want to add, I'm just telling the simplest example here. If you want to add 15 digits or some 15 numbers or 20 numbers or 50 numbers, suppose if you want to add 50 numbers together, you'll just use the calculator in mobile, in calculator, simple, and you will get the result. It will take 2 minutes alone, right? But SPSS, if you want to know mean, mode, median, frequency, chi-square, pi-square, whatever, any test, just select, click, your results are appearing on your screen. Just 5 seconds. Everything will be in 5 seconds. Thousands of the patients, lakhs of the patients, it will not take a second. It will give you the precise confirmed results. Not only add instance, these are simple things, to add, or to these things can be done anywhere, right? But for the mean, mode, median, SD, standard deviation, standard error mean, confidence interval, many things. I'll show you what else you can do. Okay, you can use extensions, Python and R programming language code directly, which are integrated in the newer versions. After 22 versions, after 25th versions, they have included Python and R programming's language codes in the open source software in SPSS. You can learn those things as well. I mean, I'm not going to teach in our SPSS because this is basics and advanced version, but this is languages I'm not definitely going to teach you. But yes, these are also associated in the SPSS softwares if you're really pro in that. I mean, I also am not aware because I'm not using the Python and extensions, but it is possible with the SPSS. I'm very honest. I don't know about the Python, how to run the Python, how to do the Python based analysis in SPSS, but it is available. And Python is one of the most language in IT and your computer science. What I have seen in the digital analytics. Okay, you can manage or select the softwares easily with the flexible deployment options. One of the very important thing is, like, if it is working with your computer or not, how to install it. It is very simple to install. You just need a key and just simple 2-3 steps. It will be downloaded, it will be installed in your computer. SPSS software, SPSS company IBM, it gives you the 100% free trial for 30 days. Okay, free trial of SPSS for 30 days. And in that 30 days, they won't restrict you to the basic or something. You will get all the access to the advanced version of the SPSS, or anywhere you can use that. 1 month, 30 days, they will give you the entire access to you, entire access. They don't restrict it to like basis or something. Even with the money, they give you just if you are paying for basic, they will give you the basic key. If you are paying for advanced key, they will give you the standard or advanced one, right? But in the SPSS trial version, they give you the advanced versions. You can use anything, bootstrapping, survival analysis, anything you can do it. Okay, with this Python programming, anything if you want. Okay, so SPSS applications, student research, like whatever research you are doing, to make it feasible, to make the results, reports, and make the publications, you need p-values, you need associations, you need confidence interval, you need standard deviations, you need standard error mean, you need mean, mode, median. Many things are there what you need to do, and those things are very much feasible with SPSS. No one is SPSS is not easy to get understand. Of course, doctoral research are dependent on SPSS. If it is a data analysis, not the laboratory work or something very specific, in the laboratory work also, this is SPSS where it is used. Every doctoral student use the SPSS version in their research. They take the help of statistician either, or they do themselves, right? Clinical trials, I'm telling, clinical trials are not using, like for the research, they use SPSS, SAS, and other softwares also. But SPSS is mandatory to perform certain tests in clinical trials. Even everything can be done in SPSS, comparisons, or I'll show you how we can do this, how it is useful. I'll show you the practical things. Okay, so clinical trials companies, they have their own. We have our own subscription. We also have at CliMed, we have our own subscription of SPSS. Even sometimes if the students need of the SPSS training course, if they need, we provide the SPSS keys for 3 months. When they are attending our course, we provide the keys for 3 months, or we provide the keys for 4 months, so they can practice during the 1 month trial. They can practice, and 3 months they can practice with our keys, and then we retract the keys, right? So it is very much mandatory to understand like how it works. That's why we provide the keys also sometimes if they require. right? And non-clinical research, as I'm telling you, in the finances, all the salaries, all the inputs, outputs, what are the like monthly sales, yearly sales, 6 month sales, everything, whatever statistics you require, you can do by SPSS. In banking, laws, management, agriculture, commerce, anywhere, wherever you need statistical analysis, you need SPSS is the best software to use. All numerical analytics can be done by this. Data management, you can do your data management also. It is very useful in the data management, and medical writing, regulatory affairs. In the pharmacy field, if I come, data management, or everywhere you need SPSS, one of the software. SAS is standard in some some streams, but SPSS is one of the additional and good certificate. Okay, so this is just the technique, just the theory part I wanted to show you before going to the analysis part. Okay, let me go to the SPSS version, and then I'll show you how to do this. Okay. Okay, so let me share the SPSS version. I'll open something, and then only I'll show you the things. Wait, modules. Okay, so let me share the blank field first. Let me open. It takes 1 minute to get opened. Okay, yeah. Okay, guys, in your computer screen now, in your screen, this is SPSS data editor window. Okay, there are 3 windows usually appears, by visual, you can see 3 windows. One is data view, where you can see all the variables, all the cases. This side, in the left side, if you look at the rows, these rows 1, 2, 3, 4 are the cases. Case number 1, case number 2, case 3, case 4, case 5, and these are the variables. Okay, at the upper toolbar, these are the variables here. Okay, in the columns. In the columns, these are the variables you can enter, and here you can enter the case numbers. These all are the case numbers. Okay, and then another view of this data, another data view is variable view. So right now, this side, these 1, 2, 3, 4, 5 are variables. Whatever variables you kept here, in the data view here, variables, it will come in the numbers in the variable. You can see. I'll show you in the data set. I have opened one data set also. So here in the data set, you will see name of the data set, type of the data set, width of the data set, decimals, labels, values, missing values, columns, alignment, measures, role. So let's see if I click here, like what type of measures it is, ordinal or nominal data. Alignment, you want left, right, or whatever. You can click and check left, right, center. Columns, you can just click on the columns and you can see what columns number you want, 1, 2, 3, 4, whatever. Okay, and missing values and labels, whatever you want to give, you can give the labels also here. Okay, and how many decimals you want in your values, 1, 2, 3, 4, you can just add on. Okay, and what is the width you want, what are the types of software, what are the types of data. So if you click on the type, there are multiple types of data, like numerical data means where you just have to keep the numbers and you want to analyze. Comma and dot means like if you have the decimals or something. Scientific notations, date, if you want to keep the dates, you can choose the any format. Okay, you can keep the dates in any format, like day, month, year, day, month, year, month, day, year, any format you can choose. Okay, year, day, or week, minutes, timings, seconds, minutes, hours, days, everything you can you can change your data sets in any way. Okay, there are multiple things. If you want to keep dollars, custom currencies, strings, like if you want to keep names, then it is see, SPSS does not allow analysis with string variables, does not allow analysis with any type of data other than numeric. Okay, so if it is string data, then it will be like only string type of data, and it will give you this description, not the specific things. So that's why that's why you have to choose, and it shows different types of softwares, what type of data it is, and it helps you that which kind of data it can be analyzed or not. Okay, it will help you in that. Okay, so this is just a data view. One thing here is very important, results window. Okay, once you do the analysis, you will go to the results window. So let me show you one data set here. I hope the data set is visual now. Data set is now visible to you. Okay, this is the data set where you can see these are the cases. Okay, this side, these are the cases 1, 2, 3, 4. So 0 here is female, 1 number codes, everything you have to be coded here. Okay, you have to give the codes, and 1 is male, 0 is female, 1 is male here. Then this in the second variable, it is age. See, columns are showing the variables, and rows are showing the case numbers. Okay, and then like duration, BMI, systolic blood pressure, diastolic blood pressure, hypothyroidism if it is yes, no. Okay, and cardiovascular problems if it is yes, no. So there are multiple things you can see. This side also, if they are non-veg, veg diets. Okay, smoking yes or no, fasting blood sugar, and you can see postprandial glyco-HB. Everything is given in the system, and in the systematic way you can see and you can analyze these things. So now what you can analyze, you got the data set, you got, and then once you analyze, you will get the result window. I'll also show you the result window. Okay, so what you are going to see, what you are going to learn, what you are going to get in SPSS, that is important. So first thing, from the file, save, edit, you can do, you can edit, you can insert any type of case files, any type of files you can import to SPSS. See, first of all, if you want to enter the data from the basics, you can enter the data completely in your SPSS. But if you have entered your data in your Excel file, okay, Microsoft Excel, then you can directly import the Excel file into this, and it will be converted into SPSS, and data will come to SPSS directly. If you have entered your data in Lotus, SAS, any type of format, you can directly export your or import your file into this, right? Okay, I'll show you, like, add the file, open the file, database. Okay, and then here you can see type of files. You can see what type of files you can open with SPSS. SPSS is very feasible with everything. Okay, you can open with you can open with the open the data of SysSat, portable, Excel sheet, Lotus sheet. Okay, and you can open the deedways. You can open the SAS sheets directly into SPSS. You can convert your SAS sheets into SPSS and analysis. You can import the data which is available in Stata, which is available in text, and which is available in SAS. Any kind of data type you can import directly and analyze in this software. Okay, and then you can view your data in different things. Now you can just convert your data. You can convert your data. You can sort the cases based on the identity, based on the variable things. You can based on the subgroups. You can just sort your data in different things. Okay, then you can sort the variables also, not only cases. You can, in the columns and rows, you can sort the cases in different parts. Okay, like from the cases, from the your rows, you can get the data and says you can merge the files. Suppose someone has entered 2-3 different files. Someone has entered in his computer, someone has entered in his computer or her computer, 3-4 friends. So you can get all the files and you can merge all the data set in together. Okay, and then you can analyze, you can run the software. So it is very easy. It is not like that, like Excel, it is not necessary to have all the variables same. Suppose you are entering with 5 variables, another person is also entering the data in the Excel sheet with those 5 variables. So it is easy to merge those type of files. But if you are merging those files in SPSS, it is very easy. Any new row or any new column can be added automatically. Okay, you can just merge it without the cleaning, or without the without keeping the keeping your variables same, keeping your titles of the column same. You can merge directly. You can split the files. You can split the files, like suppose, see, I am having here sex, okay, gender. I have males and females both in one row. Okay, one column. 0 are female and 1 are male, right? Here 1 are male, 0 are female. So I want to check, like suppose age, common, like average age, like mean age. Suppose I want to check the mean age of gender. What is the mean age of total mean age? If I want to check, I'll just go to the descriptive, frequency, I'll keep the age here. Simple, I'm just showing you. Okay. So I just want to check the mean, median, mode of the age, and standard deviation, standard error of mean. I'll just click here, is. Okay, I don't want to see the frequency table. Click, and my results window is now here. Okay, wait. I'll just check my result window. Okay, I'll show you the result window, how the result window looks like. I have analyzed that. I have clicked OK. So now you will get the result window. This is called output window. Okay, statistics viewer, IBM SPSS statistics viewer. Okay, this is the result window. Here you can see there are total number of patients, total number of subjects 63. Mean age is 49. Mean age is 49.37 years. Standard error of mean is 0.9. Median of the age is 50. Mode is 50. Okay, standard deviation is 7.56 years. So the age, if you want to write the mean age, then you can write 49 plus 7 plus minus 7. Okay, that's how we write the age. So it is just a 1 second task. It has given you the entire number, right? So, and here one more thing, without going to the data window, you can directly analyze the software, analyze the things from here. So I want to know what is the mean age of females only and what is the mean age of males only. So what I will do, easily it is not, usually it is not very easy to count, in such a way. But what I'll do, I'll go to descriptives, I'll go to cross tab. I'll just not the cross tabs. I'll go to the, if I want to check this, I will go to the, what is this? Explore. Okay, I'll go to the explore. I'll keep the age here, or I'll keep the gender here, and I'll go to the statistics, descriptives, continue. Okay. Okay. Wait, it is running. So see here, I got everything. So if you can see the descriptive, table, every descriptive is coming in the male and female separately. See, in the females, mean age is 50. In the males, mean age is 48. Can you see that? Mean age in females, it is 50. Mean age in male is 48. Median 50. Median here 48. 47 in the males. So it is very, very, very easy. Okay, let me share the data view window again. Okay, let me share the data view window again, and I'll show you what else you can do. So this is just a thing. This is just a practical thing. Okay, if you want to check BMI in male and female, BMI in hypothyroidism or non-hypothyroidism, anything you can do. Okay, so what are the software, what are the tools here you can see? What are the tools you can see are available in the analyze? What can you analyze? You can analyze parametric, non-parametric, any kind of data. You can put any kind of, what we'll say, tests, for different types of data. For example, if you want to keep chi-square test, why we use chi-square test? Like, you have the data types, different data types, descriptive data, like numerical data or categorical data, or it is usually two types, numerical or categorical, if we see. Otherwise there are other parts also of that, ranking and non-ranking data. So any type of data, whatever is available, based on the data sets only we decide what test we have to keep. Based on the data set, data type, we decide what test, where to keep, where to apply what kind of test, right? Suppose like I was checking for this mean age of genders, mean age of male and female. I was checking. So there is age is continuous data type. Age is continuous data type, numerical completely, there is no category. But gender is categorical data. Gender is binary data, right? Gender is binary categorical data. So in that, what we will use? We will either use the explore, because explore will give the entire split of the subgroups, because gender is one group, and in gender male and female are subgroups. Okay, male and females are subgroups. So you can go to the compare means also. If you want to check like what is the significant, if the gender gender age difference is significant in our population or not. Like suppose for an example, heart failure patient is like elderly disease. Elderly disease means it is usually coming in 60s of the patients, right? 60s of the years. So if I want to check, there are all heart failure patients. If I want to check whether females are younger than males or males are younger than females, right? Suppose the mean age of males are 65, mean age of females are just 58. It's just 58 years. That means the females are getting the heart failure earlier than males. Got my point? You're getting my point. Like females are getting heart failure before males. Just for an example, okay? So just for an example. But if I want to check if it is significant or not, whether my whether my results are significant or not, statistically valid or not, then I look for the p-value. Okay, p-value means the significance of the difference, significance of the association, significance of the correlation. Everything is done by the p-value. Everything is significant. Everything significant is seen by the p-value, right? So here, for the gender, if I want to check the mean age of both genders and I want to see the significance, whether the difference between male and female's mean age is significant or not, what I will do, I'll go to the comparison. I'll run the independent test. Independent test means in test variable test variable is age and your grouping variable is gender. Keep your gender here, okay? And then you can define, you can just keep like 0 for males and females, whatever, 0 and 1, continue. If I want to sign up this, okay, I got the result. Okay, I got the result, I got the plots, everything I got here. Okay, I'll show you the results window once more. Here you can see, as we were saying, the mean age in female is 50 and in males it is 48. And here you can see it is showing all the tests, Leibniz test for equality variance, it is showing the p-values, t-values, and degree of freedom. It is showing here 61, and it is showing mean difference. It is showing lower confidence interval at 95%, and upper and lower bounds of the confidence interval, everything it is showing. So in our course of SPSS, I'll take you from the basics of biostatistics. First 3-4 classes are biostatistics, where you will understand the data type, non-metric, parametric test, and non-parametric and parametric test types. What are the study designs, how to correlate, and what type of data you deal with. Okay, and then you will go to the analysis part, and then we will run the softwares, and then we will see how to do the analysis. And that kind of data sets I perform. I perform in every class. I perform the live data sets. From my PhD results, from my friends' PhD results. I give you the entire practical part. When you learn the SPSS, I give you the entire practical part when you are coming and when you are learning, right? So not only this, let's see. Now next thing is, I will show you here. In the descriptive, you can see cross tabs. This cross tab is nothing but chi-square. It will give you the chi-square test. Suppose, see here, rows and columns. Why we use chi-square? See, if our one type of data set is continuous, and other type of data set is not continuous, it is categorical, in that case we have to go for like independent pair test. But if both the data sets are categorical, suppose I want to ask, see, there is one categorical data set, gender. In that, subgroups are male and female. Another category is smokers. Okay, smokers and non-smokers. Smokers yes and smokers no. That is also categorical data. If I want to see how many males are a smoker and how many females are a smoker, then what I will do? I cannot do by independent pair t-test, because there is no continuous data. Smoker yes and no is categorical data. So I'll go for chi-square test. In that case, I'll go for chi-square test. I'll perform the cross tab. I'll perform the chi-square test. Here you can see, asymptoms. You can see statistics. I'll go chi-square test and correlations. Okay, I'll go for chi-square and correlation test, and it will give us the difference, significance, everything. Okay, it will give us everything, right? So like suppose I was telling you the example, like gender, keep the gender here, keep the smoking in column, and you can keep 3, 4, 5, any number of things, in rows and columns. I'm just showing you statistics. I just click on chi-square. I'll just show you results. You can see here, females smokers only 3, males smokers 23. In smoking, 0 is non-smoker, 1 is smoker. Okay, in smoking, 0 is non-smoker, 1 is smoking. And here you can see female in this row, males are in this row. Okay, so the difference, look at the difference. Smokers, females are only 3, males are 23. And if you look at the p-value, exact significant, two-sided, 0.000. So the difference of smoking between males and females is very significant. Males are significantly higher, in the smoker, category, or we can say directly males are, higher than females in smoking, and significantly high. Why we keep the significant word here? Because p value is less than 0.05, and the significant is shows the validation. It shows the significance statistically. Okay, that's how it is important to run the test to get the precise result. See, suppose in some communities there are, females and males are equally smokers, are equally drinking, correct? So what will it significant there? No, it will not significant there. So based on one results, like the smokers, males are more smoker than females, you cannot apply it anywhere. You cannot apply this everywhere. What you have to do? You have to perform the test. You have to perform the test in every population, every study. Then only you will come to know whether the results are significantly high or low or not. Understand? That's how statistical softwares are working and helping us, and in entire data analysis, right? Now, what else you can do? I'll just show you now because time is running. So I'll not take you. Otherwise, it takes 4 or 5 hours, and that's why we come up with the course of where we are teaching the entire like basics and advance of SPSS in 20 classes. So you can see, you can make PP plots, you can make ratios, you can make quality plots, and then you can make the tables customized or anything directly. You can compare the means. Compare the means means there are the tests, if you have heard of one sample t-test, independent sample t-test, paired sample t-test. If your both the data sets are if your both the data sets are numerical type, then or continuous type, you can use paired sample t-test to find out the significance. And then one-way ANOVA you must have heard of. ANOVA is very useful when the groups are more than 3. When the subgroups in a variable more than 3 subgroups, then we do not go for independent t-test, we go for one-way ANOVA. Okay, there are the assumptions. Every test is important with the assumptions. Okay, then you have GLM, general linear model. So in that you go for correlations, univariate correlations, multivariate linear models, repeated measures ANOVA, right? You can conduct everything here. ANOVA, MANOVA, MANCOVA, everything. Okay, and then you have GLM for generalized models also. You have mixed models. You have correlations like bivariate correlations, partial correlations, distances, regression analysis. You can do linear regression. You can do perp estimation. You can binary logistics, multiple logistic regressions. You can do any type of regression. You can do multifactorial analysis. You can do all the typical analysis you can perform here. Non-linear test, linear test, weight estimation, weight estimation, and 2G least square estimation. Everything will be performed by SPSS, and there is no coding because everything is automatically coded. You just have to select the data set. You just have to perform the data set. You just have to perform the test, nothing else. Okay, non-linear, neural network, classifications, key means cluster, two-step cluster, tree. You can prepare the, when you write the, systematic review or something, you can prepare the factors and the tree here. You can check the reliability here in the scale, reliability testing. If your scale, suppose you are preparing a scale, suppose you are preparing the questionnaire, right? Suppose you are preparing the questionnaire or scale, you have to test it in the, you have to test it in the population. You have to test it in the, your samples, your patients or anywhere. Then you have to check the reliability of that. So the reliability testing can be done by scale. The Kappa scores can be, you can get by scale or reliability testing. Okay. And then there are non-parametric tests. One sample, independent sample, related samples, legacy dialogues are there, chi-square, binomial, one sample, that anything you can do. Forecasting also you can do. Then there's survival analysis. Survival analysis is one of the typical and most advanced test. You can prepare Kaplan-Meier, Cox regression you can do. So it is like, suppose, for example, why we do survival analysis. Survival means to estimate the survival. Okay, to estimate the survival in the population. Like suppose you are following up a population for 5 years and you want to analyze that and you want to see what is the survival of cancer patients. You want to see what is the survival of, heart failure patients. then you have the data set with you. Just go to the survival analysis, apply the Kaplan-Meier, keep the years, keep the data set, perfect days when the patients died or got the outcomes based on that, and keep your variables whatever you want, and run the Kaplan-Meier or Cox regression. You will get the survival. Okay, like females are probably surviving less than males in this specific disease. You can make the conclusions, you can write the results. When you read the results, when you read any research paper, there is a result part. In that result, you see p-values, you see like the description, you see like the tables, you see the Kaplan-Meier curves, you see the different curves, different things. All are possible by SPSS. All are possible SPSS. You can go for missing value analysis, you can go for ROC curve, you can go for multiple other things in this, like okay, let me show. Okay, so by this you can see multiple and complex sample sizing. You can ROC curve you can prepare by this. And not only this, the simplest task what you usually do in Excel sheet, graphs. Prepare all the types of graphs using SPSS. Prepare all the types of graphs using SPSS. See here, legacy dialogues, if you see, you can prepare all the bar graphs, you can prepare 3D bars, you can prepare line charts, you can prepare area charts, pi square, high-low, box plots, error bars, scattered plots, histograms, everything, whatever you want, you can prepare in this, you can learn this. So this everything what we are providing, I'll just show you that. Okay, I'll just stop sharing for one sec, and then I'll show you what we are exactly planning and providing in this course, so you can understand what is exactly we are going to provide in SPSS. One sec. Okay, see, this is our upcoming course, so anyone can join this course. There is no specific requirements because this course is developed from the very basic. Course highlights exclusive SPSS learning from the research and publication purposes, and live classes, recordings available on demand, and there is 60% practical classes, complete package from data entry. Why 60% practical classes? Because the 40% of time is going in understanding the biostatistics. Okay, that type of data, type of studies, and once you understand these things, you can go through the practical analysis. This is the course content what we are completing in 2 months. Introduction to SPSS, downloading, installation of the software, basics of the statistics, definitions, parametric, biparametric, data entry, importing of the data, data computing, recoding, transformation, descriptive statistics, frequencies, data testing, all the test, independent t-test, chi-square, paired, one-way ANOVA, one-way repeated measures ANOVA, linear multiple regressions, logistic regressions, reliability testing, correlation, ANCOVA, MANCOVA, graphical presentation of data in SPSS, and then practical versions you can understand. Right. So these are all the things what you are going to understand in this. And I'll show you one more thing. I just want to show you guys. So we were just talking about SAS, right? So SAS browser is not this. Yeah. So this is the SAS base and advanced. Okay, these are the all things you want to learn. See, this is the course curriculum of SAS. Software installation, step-by-step installation, navigate, work in SAS environment, learn how to get data from external sources, create temporary and permanent SAS datasets, and then combine datasets, data and time functions, MDI, YDR, core, month, day, weekly, today, everything. Conditional statements, other statements, logical and conditional clauses, attributes, databases, length, label, everything, data set, step, revive, and these all everything you are going to learn in our SAS course, guys. Okay, so with this I think we can create we can actually complete this, and if you have any kind of questions, you please tell us, and you will get all the links in your email. Feedback link I'm just sharing with you. in the YouTube as well. So you can fill the feedback, and you can, and if you have questions, please go ahead with the questions first, guys. Okay. So this is the feedback link. This is the feedback link I have shared in the YouTube. Okay, questions I'll take now. And registration link for the SPSS and SAS is available in our description of this video, and you will get the email about that also. Okay, if you have not seen the video, you can see there also. Okay. Yes, yes, softwares are available for the MacBooks also. Yes. Which one we will choose, SAS or SPSS? Both are distinct. SAS is, like if you are going to, both are applicable in the industry also. Wherever you want, both have this, like distinct things. SPSS is, more valuable in, data sets, data management, and those things. And SPSS, I will say, I'll be honest with you, SPSS is more into the clinical research, medical writing, and those things. And SAS is, SAS is everything actually. You can do that. Is there any opportunities for freshers with respect of SAS program? Right now in our company, Sandhya, no, we don't have any type of vacancies right now. Not in our this also CRO also. But yes, we can recommend, once you complete the course with us, we can recommend you to the industry, other industries, and by that time, if we get the openings, you will be placed in our company only. But there is no guarantee, because this is totally the educational program, not the assistance, not the job assistance. How can trainee do practice as they don't have SPSS installed? We are, helping them in installing SPSS trial version first for 1 month, and for next 1 or 2 months, we are giving the keys from our side. Our, side for 2 months or 3 months, we are providing the keys so they can practice while learning. There is no issue. SPSS keys we are providing once they are learning, only for 3 months. See, standard error is a precise, difference between standard error is, the next version, I will say, of standard deviations, because standard deviations sometimes cannot work, and if the standard deviations are not matching, we don't go for means. But standard error is something more precise, which gives us the difference between the characteristics of two variables or two data set. I mean, two variables, of course. Why? When we are comparing two variables, sometimes standard deviation is also similar. Okay, but when standard deviation is also similar and all the characteristics are same in two variables with the same number of sample size, with the same number, with the same standard deviation, same means, in that case we go for standard error of mean, because standard error of mean gives the actual error term. Okay, it gives the actual error term, and that's why it gives the precise results, like what is the actual difference between two data sets which looks similar. Suppose like there are two groups of students. I want to go for, I want to check the mean age or mean weight of all the students, right? But their mean age is similar, like both the batches, or both the sections of 10th class, 10th A and 10th B, if I want to compare both classes, shows their mean age is similar, mean weight is similar, everything is similar. But how to get the difference between those? So then we go for the standard error mean. What are the similarities and differences between SPSS and GraphPad Prism? Okay, Subhecha, see, GraphPad Prism is actually, I do not know much about the GraphPad Prism, frankly, because I have never used, but I have gone through the workshops on GraphPad Prism also. So there is no much difference, but GraphPad Prism is usually not for the typical analysis is like survival analysis, or like the regression analysis, multiple regression analysis, or ANOVA. Those are tough, and there you have to go for the specific codings and specific codes with that. SPSS is more, I will say, is user friendly and gives you the comprehensive results and comprehensive analytical things. Okay, Anshul, what space basically is significance? Significance tells you about the difference between two, variables or two things. Suppose you are seeing like, from the far, suppose this is a basic example, what we give in statistics. There are two things. One is cow, a small cow baby, and or, one is a full tiger, but it is far, you cannot completely recognize. Okay, you are just looking at the images and you are not able to recognize what is the difference, right? Same thing with the data sets, because data sets are like a blind thing. There are thousands of the sample sizes, thousands of the entries. You cannot make the actual difference. You look at the numbers will not tell you the difference if there are thousands of the data sets, right? Thousands of the sample sizes. In that case, what you do is you perform the analysis. You get a binocular to see that images. Here you get the software to look into the you dive deeper into the data sets. Okay, once you see the data set, you will get the p-value, which will tell you like what is the exact difference between two data sets, why these data sets are different, and the p-value says the significance between those, like, okay, this data is significantly higher or lower than other one. Yes, Jahir, you can join that, because all the classes are through the Zoom only. There are the classes Zoom. Duration of SPSS course is 4 weeks. No, Siddhika, see, our SPSS course is in the weekend only. We conduct in weekend or in the evening hours after 6 p.m. only. I take classes because I also have offices, I also have work. So I take classes only after 6, only after 5 or 6 in the evening. So there is no worries, and definitely we are providing recordings also. If you miss the class, you will get the recording of that. Oh no, SPS and SAS are not for the mobile, Sonika. These are not mobile softwares. Not at all. No one is mobile software. You cannot, download any of the software in mobile. Yes, SPSS makes a big difference. Khushi, it makes a big difference in getting a job in clinical research organizations because it's very useful. In clinical research, see, first of all, I'll tell you, if you are publishing a data, if you are publishing a paper, you need analysis. You need to put the results. And in the CRO, everything, everything relies on data sets, data management, data publication, medical writing, regulatory affairs, anything. All the departments are relying on the data. And if you have the data, you need SPSS applications or SAS applications. Mostly, if you have the SPSS knowledge, you will get the advantage, the significant advantage in getting job and even in your promotions. Anyone can download SPSS software for a month free of cost from the IBM site, but it is only free for a month. And if you think it is email based like people do with such softwares and all, like they put one email for 1 month, then another email for 1 month, it is not like that. Once you install SPSS in your computer, it will be, if you delete also, if you uninstall also, it will be getting that IP number. Until unless you do some specific techniques to remove out the things, it will not accept. Your computer will not accept. After trial version, it will not accept with even different email IDs. There are some formulate that we need to remember, not inbuilt in SPSS. No, I don't think so. There are very less things which you have to remember. You just have to remember assumptions. Veena, that is mandatory to read the assumption and understand the assumptions. If you don't understand the assumptions, if you don't remember the assumptions, you will fail in SPSS, doing SPSS, because every test has their assumptions. Data sets are similar, data sets are same, but how and where to apply which test, that is what you have to learn. Assumptions. Yes, chi-square test can be used for both ordinal and interval data. Yes. Yes, we are providing the university edition of SAS, Kavita. Yes. How much days for certification? See, after the course, see, yes, both the course, guys, it is very much mandatory to tell you, both the courses have exam at the end. If I teach you, because these are my company and our programs are under the NEP, new educational programs, new educational policies of Government of India. So if you are providing the course, you have to write an exam at the end of the course. That is easy, multiple type of questions, but you have to clear the exam to get the certificate. Okay, and that is easy. That that is very easy, the exams and all. You will get free for 1 month. I mean, the trial version you can download from IBM, and then next 3 months we will give you free of cost our paid version key. We generate the keys for every of our student. SPSS is very useful in pharmacovigilance because pharmacovigilance depends on the data reading, data understanding, p-values, significance, R square, adjusted R, adjusted survival, everything. Next batch, we are not sure. But right now, the batch is starting on 7th May. You can join both the courses at once. Not a problem at all. Because classes are not, together. Classes will be either before or either later, but classes are not, classing. If you really want to learn right now, you go for, SAS right now. You go for SAS. And SPSS you can learn in the next batch. If you have zero knowledge in biostatistics, you can join the course, of course, no doubt, because we are teaching from the basics, very, very basics. From the downloading of the software, how to use, what to use, and what are the importances, everything we are taking from the scratch. Okay, guys, then if no more questions, then we'll take off and you will get the email around 5:15 ml. 5:50 p.m. and you can fill the if you want to register, you can register from here. But remember, today is the date where you will get the these discounts. Otherwise, you will not get. Otherwise, you have to pay the entire fees. No, see, from our batches are not very consistent, not every month or every 2 months or 3 months. Our last batch was in July 2021. After that, this batch is now in the April or May. So we are not very frequent. Our next batch may come in November or may come in the next year, 2023. We are not sure. If you want to join, you have the opportunity today. SAS program is 3 months. SPSS is 2 months. SPSS is not exactly 2 months. I will try to finish it in 1 month because people, what I have experienced from the previous batches, the people, if we are taking the every week classes, they are not, sometimes they are not able to connect the dots because there are many things. It's tough to read the assumptions and all. So I'll try to complete early. I'll take the more classes. Complete early means right now we are fixed for weekend classes, but sometimes what I do is I take the classes in weekdays also if it is feasible for the students, only based on the students' convenient. Okay, guys. So you think of, take care, and see you. Bye. Good night. Thank you for. Good evening. Thank you for attending the session. Very nice to see you all. Thank you. Fees for SAS, if you are joining today, is 5,099 only. 5100 rupees. See, I'm sharing again the links, and you can see in the description of this video. This is the fees for. This is the link for SAS. This is the link for SPSS. Okay. Take care, guys. Chalo, we'll put off the classes. Thank you. What is the difference between SAS and SPSS? Jacob, we are describing this since 2 p.m. What is the difference between SAS and these? Both are statistical softwares. SAS is quite more used in the data management, data analytics, and CROs. But SPSS is something which we need at the basic level, which we need at the research level, and for the clinical research, as I'm from the clinical research background. But it is useful for all the streams at the basic level. And once you are going to Pro 1, you can definitely go to the SAS programming as well. You need SAS programming also. Sure, anything is fine. Thank you, Shreya. Thank you very much. Okay, take care then. I'll just put off the things. Thank you, guys. Thank you. See you. Take care, everyone. Thank you, Dr. Tarun. Thank you for patient listening.